Hi there. So I'm Matthew, and today I'm going to be talking about this paper. It's Protein Stability Engineering Insights Revealed by Domain-Wide Comprehensive Mutagenesis. Kind of a mouthful. We'll talk about what all that means. So just a little bit of overview of what we're going to discuss in this presentation. First, we're going to talk about what exactly is the main takeaway from this paper up front. What are things to keep in mind as we go through all of the sort of methodology and the figures of this paper. We'll talk a little bit about the relevance. We'll go through the methods that they developed. We'll look at the actual experiments they performed and the comparisons that they did to current methods that are out there for evaluating mutagenic effects on proteins. And then lastly, we'll talk about what are the main takeaways from this. What can we apply from this paper to our protein engineering practice? So in this paper, they demonstrated a high throughput experimental method for investigating protein stability versus primary structure. Essentially, they're making single amino acid changes in a protein. They're going to look at how that affects the overall protein stability. And they're able to do that for the entire primary sequence, basically. And not only that, they're also able to evaluate changes for every amino acid substitution in every one of those positions. And then it's also a sort of comparative paper. We're going to take the results of this experiment, because we have real experimental results of the stability change from doing all those mutagenic changes. We're going to compare that to computational methods that are used to predict protein stability versus primary structure, and how we can improve our prediction methods based on the information in this paper. So I think we all know protein stability arises from the primary structure, and it is like the critical statistic in the context of protein engineering, right? If you have a protein that's stable in physiological conditions, it sticks around for a lot longer, it is hard to digest, it might be favorable for ligand binding interactions. There's kind of an open problem, which is how does the substitution of a single residue impact the stability of a protein? And there's a lot to think about in that, right? If you make a single residue substitution, is that going to disrupt any salt bridges? Is it going to disrupt any H bonds? What happens to the interactions between the solvent and the protein itself, right? If you're exposing hydrophobic residues to the solvent, is that going to cause like clathrate disruption? Is it going to solvate worse? And conversely, like what happens if you place a polar residue facing towards the inside globular part of the protein, right? All that stuff is sort of hidden away from the solvent and it's non-polar, so you're going to have a huge energetic impact there as well. And then there's also expansion or restriction of the conformational landscape. You were talking about glycine, how glycine is really flexible, right? Protein's really rigid. Both of these things have impacts on the stability and the structure of a protein in terms of its backbone and the allowed conformations it's able to adopt. So for this reason, because there's a lot of parameters to think about, right? And you can't just sort of theorize about it. In silico approaches are used to predict the impacts of altering residues. And there's a quote from the paper which really states the kind of current situation of in silico methods, which is that they can capture general trends, but they fail to predict the magnitude of mutational effects. So they're not so good. And there's varying degrees of not so good based on the structure that you input or provide to that in silico method, the conformation sampling that method is able to use in terms of, like, is it doing any sort of energy minimization or anything like that? And then, of course, the training or testing data that method was developed with. Some of these have training and testing data that's rather restricted in size. The alternative to predicting things in a computer is, of course, getting the values experimentally. And there's a couple ways to do that. There's calorimetry of all the variants to assess the protein structure. There's also spectroscopy of the denatured variants, and that's what ends up being used in this paper for the experiment. And you can denature those proteins either chemically or thermally. They choose to do it chemically. What quantities do you actually get out of one of these assays? So you get what's called the denaturation midpoint, and here in this paper it's going to be the chemical denaturation midpoint, the CM value. You get the change in free energy of unfolding, the literal change in energy of a single protein unfolding. And then you get the change in free energy, this delta delta G value. And that tells you what's the difference from the wild type protein to the variant protein in terms of the change of free energy that you observe between those two. So there have been a lot of measurements over the years for several proteins and variants, which the computational models I talked about earlier have been trained on, right? Like they've compiled all of these into a big database, and then you're able to stick a computer on the database and develop a computational method using that. The problem with this, though, is that experimental databases are gathered from a vast set of disparate experimental conditions, right? Things are not always performed in the same way for every experiment or value that gets entered into a database. And so the data quality is a major factor in the quality of the model that's produced. Right. So here's the big question, right? Is there a reasonable method for consistent whole domain stability analysis of residue substitution? And that's what they perform in this paper. That's the experiment that they do, is they're able to have one experiment, one assay that's able to go through the whole set of substitutions that are available and do stability analysis. So how exactly did they do that? So they selected a moderate-sized domain of a protein to analyze. And in this case, it's the beta-1 domain of a streptococcal protein G. It's 56 residues long, and there's a lot of secondary structure. There's beta strands, there's an alpha helix, there's coils. So lots of secondary structures to look at in that domain. And what they did is they developed a plate-based method for stability determination. And then they generated 935 variants using genomic engineering, and then were able to assess those for each variant, the stability for each variant. Right, so after that, they took that data and they were able to analyze the positional sensitivity and the residue impacts from that data. And then we were able to compare it to in silico techniques that are out there, as well as this deep mutational scanning thing that I don't really quite understand, but they compared it to that as well. So here's a sort of diagram of what they did. They first change the genome of that G beta-1 domain and generated all of the possible mutants, single substitution mutants for those, right? So they were able to hit 17 of the 19 possible residues. They avoided tryptophan and something else I can't remember right now. You'll see why. And then they were able to substitute in 55 of the 56 positions on that domain. They were able to then culture those modified streptococcus and then have them express the G protein, isolate off the G protein, and then they used that isolated product in a, quote, previously developed plate-based stability determination method, which if you go and read the supplemental information, essentially what they're doing is they're measuring the tryptophan fluorescence as a function of a denaturate concentration. And so what you generate as a result is this curve. And the slope of this curve, the transition between those two states, as well as the midpoint of that transition, ends up giving you that delta delta G value. And if you're curious, it's the midpoint of the wild type minus the midpoint of the variant times the average slope between those two fluoroscopy experiments. So what did they actually do with this data? So this is the presentation of these in the paper is not great. The first three are sort of all related to each other, so I thought that I would just present them as they're presented in the paper. This first figure is a heat map which indicates the changes in stability as a result of the particular substitution and the position of that substitution. So most of the variants result in a neutral change in protein stability. It's not really demonstrated very well. Their color scale is very quick to pick up color here, but about 60% of the variants here, 68% or so, are neutral and don't really impact the stability that much. Something to keep in mind, these delta delta G values, if you come from like chemistry world like I do, you think like negative delta G is spontaneous, and right, so negative delta G is better. That's not how this works. Positive delta delta G is better here, and negative delta delta G is destabilizing. So the negative values are in red because those are the ones that destabilize this whole system. And the reason for the sign change is because we're tracking the process of unfolding, not unfolding. So like I said, about 68% of the mutations are positive or neutral in nature. They talk, they mention that might be an overrepresentation compared to proteins or specific domains in general due to the high surface to area ratio in this particular domain that they selected. And if you look at databases that are out there, they say about 50% of mutations are destabilizing. So it might just be the particular domain that they selected might have an impact on this. So one thing to notice from this is that there's a horizontal trend here, right? So along here we have the sequence of that G1B protein, and then horizontally we have what residue did they actually substitute into that position. And you can see that the only really vertical trends are when you make a substitution with a glycine or a proline, because those, they introduce conformational changes that will throw off the stability of the protein. But most of the trends are horizontal. And what that really means is that all of the sensitivity to stability impacts are positional in nature and not really polar in nature, right? It doesn't really matter so much what residue you're substituting into the protein. It matters where you're substituting it in the sequence. Right, and the big thing here is that all of these black squares are so destabilized that the protein just can't fold or it's not expressed. So here's sort of a different view of the data that we just looked at. In part A, this is just a distribution of the stability change across all of the variants that they have. We can see that the greatest area is sort of centered around 0. So most of the changes are neutral, some of them are stabilizing, and there's sort of this tail of destabilizing variants that are in this negative range. And that includes the qualitative set. They couldn't get any numbers off the qualitative set because they just were unfolded, couldn't do the spectroscopy. If we look at part B of this figure, what they did is they classified the variant change in as to what tertiary structure that change was performed in. So you'll see they have a coil, or core, boundary, surface. If they're making changes in the core, you can see from this diagram that those are more often than not going to lead to qualitative data as opposed to quantitative data. So changes in the core are really destabilizing. More so than changes at the boundary or changes at the surface. And so then they asked, right, can you do this analysis on the secondary structure as well? Like if you're looking at what is the proportion of changes or what is the impact of stability when you're looking at loops versus helices versus strands, is there an observable shift in the stabilization at the secondary structure level? And no, there's not. You can see the median and the interquartile ranges are pretty consistent across all of those secondary structure categories in part C of that figure, in that middle box. And you really only get that big difference in the tertiary structure, right, if you're doing that tertiary analysis. And for the core, the median shift for core residues of delta delta G is negative 1.5. So that's pretty extreme considering the context of, or relative to what's happening in the rest of the paper here. This is not anything new. We're engaging the visual cortex here. This is just a really nice visualization of what's happening. We are mapping that median delta delta G value that we just saw onto each position in the protein. And it's really obvious here that the positions that end up with the greatest destabilizing effect are all on the interior, and the ones that have a stabilizing or neutral effect are all on the exterior. Yeah, okay. So one thing I will say is that not all of the changes in the core are the same. There are different degrees of destabilization that happen there. And they make a big point of discussing this in the paper, is this core residue sensitivity. So they looked at what sort of attributes in the positions that they were making changes at, and also the amino acids that they were making changes with, to see if there was any sort of predictor of replacement stabilization effect, whether if you're replacing something with a hydrophobic residue, or a really big residue, or something like that, whether the difference in the values between the original residue that was there and the new replacement, whether you could sort of predict whether or not it was going to have a destabilizing effect. And here are some of the attributes that they probed. They looked at the change in volume, change in charge, change in flexibility. Of those, they found that the best predictor of behavior were the measures of residue burial, so whether or not something was in the core. And so they're looking at the depth of the beta carbons for each of the residues, and then also what's called the occluded surface packing, which is just it's another measure of how deep or entrenched residue is in the context of the residues around it. So those both had correlations of 0.82 and 0.76 with respect to the change in delta G. So they were pretty good predictors. They also looked at combinations of different attributes. Those didn't do any better. Really, the best two were looking at the depth of the beta carbon and the occluded surface packing. So now for something completely different. This is going to be interesting. So then what they did is they ranked the amino acids according to the worst tolerated and best tolerated in the figure. And essentially, across all of their variants, right, they took all the ones where they replaced something with a, glutamine, and put them all together and looked at what the impact was to the delta G, or the stability, from making a change with that amino acid, right? And they did this for all the amino acids. And then they ranked them from the worst tolerated to best tolerated. Right? And tolerated here just means stabilizing across all the positions in the sequence. So what were the best tolerated residues? There's a lot of surface, we already mentioned that. Might surprise you, they're all hydrophobic. Isoleucine, phenylalanine, those kind of take the cake for terms of stabilizing residues here. Something interesting to note, when comparing polar or charged amino acid pairs, the more nonpolar member is preferred by the rankings. So if you look at asparagine versus glutamine, right, Q versus N, you can see that Q is better tolerated than N, which is all the way sort of in the middle of the figure. Same with aspartate or glutamic acid. So that extra methyl group, that more nonpolar nature of those even charged or polar amino acids, contributes to the stability. What were the worst tolerated residues? Hydrophilic with high charge. And aspartic acid is a good example of that. Here they sort of break things down in a little bit different analysis. So we were looking at tertiary structure before, we're going to look at tertiary structure again. When breaking into positions of change in regards to tertiary structure, hydrophobic residues are better tolerated in all categories. So it doesn't matter if you're looking at the surface or at the boundary, hydrophobic residues still win out, which you wouldn't really expect. Something that's also very interesting is that they looked at the size of the hydrophobic residues they were replacing with. And the greater the increase in surface area and volume, the better. That doesn't make a lot of sense. You can look at where isoleucine and valine are on this chart. It's kind of in like the bottom of each of these green ranges here, compared to the larger hydrophobic amino acids. So remember that this is all in the context of single replacements. It's likely that if you were making multiple replacements, this would not hold up. They make a mention of that caveat. Polar residues are relatively better tolerated on the surface, but still worse tolerated than hydrophobic residues. So I just thought this is really interesting. It kind of goes against your intuition. You think that, like, in physiologic conditions, the solvent's water, right? And so you would think that polar stuff would be pretty happy to be on the outside, would solvate a protein better, make it more stable, but that happens to not be the case. So then they took this data and they compared it to the computational methods that are out there. And they were looking at, well, how well do those models perform in recreating all of the experimental stability changes that we just measured? Um, and is there any way to improve or use those computational methods more effectively? So there's sort of like three big models that are out there that they looked at. There's one called Pop Music, there's FoldX, and then there's Rosetta. So first they looked at the data sets that were used to produce these models. You can sort of see if you read through this table, the main takeaway is that the FoldX set is a lot smaller, less diverse. Pop Music is the only one that's definitively better. It has like double the number of measurements as this experiment. Rosetta is like comparable. All of the data sets heavily emphasize like alanine scanning, mutagenesis, alanine variants, because that's a pretty standard thing to do when you're doing these sort of protein stability analysis, as far as I know, is replacing with an alanine because it's small and nonpolar, and so it's kind of like a neutral way, so you can look at the change in free energy there. And so that's what a lot of these data sets have been compiled with. And you can see that the percent alanine for a lot of these variants that are in these data sets is pretty high compared to the percent alanine replacements that were in this data set that was generated. Interesting things about the computational models. So Rosetta allows for some parameters as a part of the prediction process. You can specify things, whether or not to perform energy minimization, whether or not the backbone has some sort of constraint on it. It's the best performer out of all of these. Even then, the best performer is, the correlation's not great. It's like 0.6 based on when compared to the experimental data set. So it's not great, but it's the best performer of what they have there. So how do people actually use these computational models in practice? Well, they mentioned the most common strategy is so you generate stability info for all of the mutations of a domain, and then you filter by some sort of stability cutoff, and then you can look at the variants that sort of make the cut, that make the stability change that you want, and experimentally verify that those are the best performers, and then do something with those best performers. So which computational model is the best at this strategy when compared to the data set we looked at earlier, or that we generated in this paper? And that leads us into this figure. So this is a little convoluted. They wanted to look at what is the predictive power of a model when compared against this data set, to come up with a number of variants that were surpassing their cutoff, or the number of variants that they wanted for the stability change, as the number of hits that they allowed, or the number in that cutoff, increased. So essentially what they're looking at is like, I ask this model to give me 20 variants, or like my cutoff includes 20 variants. How many of those actually give me a positive stability change that are a true positive, that are not a false positive, not a false prediction? And that's what that PPV value is on the y-axis here. The performance of these is really context dependent, is really the takeaway from this. The best performer overall, they mention, is the combination of looking at the output of Pop Music and Rosetta with no energy minimization. When you're looking at part B there, that's just for reference. It's the ranked stability change in terms of they made so many variants. Here's how many have a positive stability change on the y-axis. So then they looked at this other technique for coming up with protein stability predictions, and that's deep mutational scanning. Don't ask me anything about this because I don't know. The only thing I know is that it allows for evaluation or scoring of a wide set of variants based on genotype to phenotype mappings. How they make those mappings, it's kind of murky to me. I'm not someone who's very good with genotype stuff. So what they did is they looked at current literature that was out there that made some predictions using deep mutational scanning of this specific domain, of this G1 or G beta 1 domain, and they compared that to the experimental data set. And that's what you're looking at right now. And that was from Wu et al. And you can see the correlation here is, again, it's 0.6. It's not any better than the other computational method that was employed. They talk about some reasons for why that is. Alanine substitution is still favored in deep mutational scanning because alanine substitution is really common in terms of like things that have actually been genotyped, as far as I understand. This is a really poor, you can see these orange like core variant mutations are like all over the place in this figure. So really what this is saying is that this method of coming up with energy values from deep mutational scanning is a really poor predictor of core residue change performance or stability change. They looked at another way of calculating energies from deep mutational scanning. This one was from another piece of literature called Albanowski. Better performance here, but still it's still poor correlation, right? 0.72. You would expect that this method, because it does like a genotype to phenotype mapping, might have a better correlation than some of the computational models that just look at energy minimization. But it's still kind of a poor performer. So that's the end. That's the end of this paper. So what are the main takeaways? So they came up with a method for doing high throughput protein stability analysis, and they used it to analyze this particular domain. The one I thought was really surprising, it was kind of neat, hydrophobic single substitutions are extremely well tolerated, and they aren't going to break up your protein or make it significantly less stable. And the biggest factor in terms of determining stability change is the position of where you make that change at in a protein or in a domain. Computational DMS methods are useful, right? It's cheap to run a computer, but they could stand to see improvement, and that improvement is going to come from developing methods with larger, more consistent data sets. How can you generate larger, more consistent data sets? You can use the technique that was outlined in this paper, where they sort of do all of this well plate method for protein stability analysis. That's it. Any questions?
Yeah, so I was wondering, like, what were the special amino acids? I think it was in purple on one of the graphs, and why would they label it special?
So if I remember right, oh my word, I have to go all the way back here. It's glycine and proline.
Okay.
And it's just because glycine's so flexible, proline's so inflexible, they're going to mess up the backbone of what you're looking at. And so it's just going to destabilize anything that you insert them into, right? I remember when I took biochemistry, my professors, oh, is it forward?
Yeah.
Did I pass it?
Yeah, you passed it. It's there.
Yeah, it's glycine and proline, PG. And it's the same in figure 2, which is what I thought you were talking about, the ones over on the far right. They also label those as special as well. Anyway, the same, my biochemistry professor called proline, it's the destroyer of alpha helices, right? So the conformational backbone stuff has a huge impact on stability, as I understand it. Anyone else? No? Okay. Thanks, everyone.